All right, thank you, everyone. My name is Stephanie Eisenbarth. I'm at Yale University in the departments of Laboratory Medicine, Immunobiology, in the section of Rheumatology, Allergy, and Immunology. And I'm going to talk to you today about some of the work that's been funded by FAIR. So I'm going to start by just giving an overview of how we are approaching the study of food allergy. The overarching question for most of us is, how are we going to find new approaches to both the diagnosis and the treatment of food allergy? And my bias in all of this is that by understanding the fundamental immunologic pathways that promote food allergy, we will be able to identify new, unexpected, potentially ways of intervening in this process. So the story I'm going to tell you about today is a continuation of one that I've talked about over the last couple of years at the FAIR retreat. You can see down at the bottom here on this slide, obviously we're well aware that there's numerous allergens, food allergens out there, with some dominant ones in the middle. I think the other thing that everyone is quite well aware of is that food allergy is clearly a combination of both environmental factors and potentially some genetic factors that set you up, unfortunately, for having this allergic reaction, whereas most people don't have this reaction. And so one way that we have tried to approach this is by using some of the genetics that are known to try to understand monogenetic forms of food allergy, to see how those teach us about what the pathways that are potentially acting in a healthy person, but then go awry and develop susceptibility to food allergy. And as you can see, there's a number of genetic loci that have been identified. We're going to talk today about Doc8 deficiency. This is the system that we have used to try to uncover new pathways and new understanding. So Doc8 is a molecule that's operational inside cells. It's a guanine nucleotide exchange factor. It's involved in signaling. It's involved in the actin cytoskeleton. It does actually quite a number of things, and it's in many cells of the immune system. So it's quite pervasive. But the reason we became interested in it is because there are a very rare group of patients with mutations in Doc8 that actually present with an immunodeficiency syndrome, but also a paradoxical allergic phenotype. And so they are a group of syndromes called hyper IgE syndrome, or HIES. And this grouping of diseases presents typically with an elevated level of IgE. And so that's often how patients are picked up. But what's unique about the Doc8 form of hyper IgE syndrome is that they also present with very severe food allergies. And that's slightly different than the other forms of high IgE disease states. And so many years ago now, we reached out to FAIR to help support us to understand why Doc8, what it was doing, and why these patients were presenting in this way, with the idea that we could uncover fundamental aspects of the immune system that were operational again in non-Doc8 mutated patients that were leading to food allergy. And so we did this by developing a mouse model of Doc8 deficiency, one in which we could delete it in particular cell types. And we've made a number of discoveries over the years, which I have shared at different retreats. So I'll just summarize at the beginning and then get to the new data. One is a story that just came out last year, in that when you lose Doc8 inside a T cell, it actually drives those T cells to become a weird, very unusual cell type, which we call TFH13 cells. These are cells that go to the B cells in a particular area of the lymph node and produce an unusual set of cytokines. And so these cells we found by deleting Doc8 in T cells. But what was really important is that we were able actually to find these cells in patients with peanut allergy that did not have Doc8 mutations, and in mice, wild-type mice, that also did not have Doc8 mutation, but were susceptible to food allergy. And so this helped us uncover, again, a very fundamental pathway, which is diagrammed here at the bottom, in which this type of T cell produces an unusual set of cytokines and directs a very specific type of B cell to make IgE that's capable of inducing mast cell degranulation and anaphylaxis. And again, these cells were there in wild-type mice and patients with food allergy, but they were so rare that we were not able to find them until we used this system where we had deleted Doc8, and then those cell numbers expanded and we were readily able to detect them. And so this was one important finding that we are now following up on in terms of understanding how this actually happens when Doc8 is sufficient in a mouse. How do we get these TFH13 cells? Similarly, we've done work, again, using these mice, deleting Doc8 in other cell types to show that dendritic cells are also impacted when Doc8 is lost, and that affects the type of T cell that's induced. And finally, more recently, work that I presented at last year's retreat, Doc8 is also operational in B cells, and there it's important for the B cell response to food allergens in the gut. To make a very specific type of antibody, IgA, so different than the IgE that we know is important for inducing the symptoms of food allergy, like But here, this is directing a different type of antibody, IgA, which we really don't understand its function. But we know that Doc8 is important for that B cell response. And so we're trying to understand both how that at the molecular level happens, but also try to use this system to understand really what IgA in the gut is doing. So I'm not going to talk about those stories today. Instead, what I'm going to focus on is a story where, again, Doc8 has been instrumental, but not quite in the same way as those stories I just showed you. And that was how Doc8 might be regulating how food allergens are handled in the gut. The way that we started to explore this was by focusing on a very particular mouse strain that has been shown to be highly susceptible to food allergy, and that's the C3H mouse. What you're seeing here is a family tree, essentially, of all the inbred strains of mice that have been fairly well characterized. Things like B6 mice, which are up at the top, BALB/C mice, which are a typical model for like allergy, like asthma models, NOD mice, which are diabetes model. And then down at the bottom in the red box are the C3H mice. And actually, these mice are not a very common mouse strain for the study of immunologic pathways, except for one very notable exception. These mice have a spontaneous point mutation in the molecule TLR4. Toll-like receptor 4 is a part of the innate immune system. It's a pattern recognition receptor that is very important in detecting gram-negative bacteria. And this mouse strain was actually one way in which this important molecule was discovered, and therefore has been a model for understanding the innate immune system. But outside of that, it's not a typical mouse strain used for other types of studies. So it was notable that C3H mice were one of the primary models that multiple groups had shown were really susceptible to food allergy, in particular peanut allergy. And we wondered about this in particular because we had discovered a number of years ago that these mice are actually the only one on this tree that have a spontaneous point mutation in Doc8. So we wondered whether or not Doc8 might be relevant to the phenotype of these mice in being highly susceptible to food allergy. Now, I will note, though, and we'll come back to this point, that actually the role of TLR4 in food allergy is also important to consider, as this molecule is expressed in the gut and is important in terms of regulating bacteria. And both of those types of responses are obviously important to the gut homeostasis in response to food antigens. So that had been one of the leading theories out there in terms of what might be the susceptibility locus in these mice. However, there'd been multiple papers published on this, not all agreeing about whether or not that was true or not. So we're going to come back to that point. But just to begin, we started by first just characterizing one of the most common mouse strains, inbred mouse strains, the C57 black 6, and comparing it to the C3H mouse in a peanut allergy model that we and many others use. And this involves oral administration of peanut in the presence of an adjuvant cholera toxin. And when you do this, it induces a very robust IgE response to peanut. And in C3H mice, it also can induce an anaphylactic-like response when peanut is given as a challenge. So what you're seeing here in all of the red squares are C3H responses, and in all the blue circles are B6 responses. And we're using a number of readouts to assess and gauge the anaphylactic response. First, on the upper left, somewhat unique to mouse strains, we can use temperature drop as a readout for anaphylaxis, and you can see only C3H mice really respond to this oral challenge with peanut. Similarly, we can use a clinical scoring system for anaphylaxis, and again, the C3H mice really stand out as quite different from the B6 mice. And then the bottom left, we have MMCP1, which is a molecular marker of mast cell degranulation, so more of a molecular readout of anaphylaxis. And again, only the C3H mice are responding. This is in contrast to one of the most important susceptibility factors for this allergic reaction, the IgE level. So we can measure peanut-specific IgE in the serum, and indeed, there's really no difference between the B6 mice and the C3H mice. So this argued that the allergic induction response was not different, but that something about the actual response to the food antigen, potentially in the gut, was actually different. And so we wanted to address whether or not there was a genetic cause for this in the C3H mice that we could identify. As I mentioned earlier, at looking at TLR4 because of its function potentially in the gut. And we did this by restoring TLR4 function genetically in the C3H mice. This didn't alter peanut-specific IgE, as you can see in the bars graph, but importantly, it also didn't alter the anaphylactic response. So again, now we're looking at an anaphylactic temperature drop readout in the mice over time, over the course of 45 minutes. And again, you can see the red line and the blue line really overlap, so they're not different. Similarly, the scoring system and the marker of mast cell degranulation, MMCP1, just really aren't different. So this argues that TLR4 really isn't the genetic cause for the susceptibility of this mouse strain. So then we turned to Doc8. And again, we did the same thing where we genetically corrected Doc8 and its function in this mouse background, the C3H background. And again, it did not alter peanut-specific IgE, but again, it did not impact the anaphylactic susceptibility of this mouse strain. The temperature drop was intact, the anaphylactic response was intact, and the MMCP1 was intact. So these data rule out these two genes as being highly likely as the cause for the susceptibility of this mouse strain. We turned to really trying to deeply characterize what was going on potentially in the C3H mice as compared to a mouse strain that was resistant to this anaphylactic response, the B6. So kind of returning to the data I showed you at the very beginning. So one thing we tried and had been proposed out there in the literature is that actually it depends on how you administer antigen for the oral challenge, in this case, how you give the peanut to induce this anaphylactic response or not. So in the left-hand side, you can see that when you give antigen orally, or PO, you can see this huge difference in the change in temperature and, as I'd already shown you, changes in other parameters of anaphylaxis. But instead, if you give peanut intraperitoneally, which bypasses the gut and it gets the peanut systemically in the mice, then the response looked the same. So this is consistent with them both having an intact IgE response. But that argued that there was something very specific about the gut response to the oral challenge with peanut that might be impacting the susceptibility to anaphylaxis. And so we used a trick that's out there in the mouse systems that we can administer a fluorescently labeled molecule, phytidextran, and we can look to see, is it getting absorbed systemically? So going in through the mouth and coming out into the bloodstream. And indeed, we saw a difference when we administered phytidextran orally between B6 mice and C3H mice. And the C3H mice were absorbing more of it. And so that was an artificial but very useful tool, first identifying this. But we subsequently used another anaphylactic readout or allergy readout called passive cutaneous anaphylaxis, or PCA. And so what we do here is we take serum that has IgE, in this case to peanut, and we administer it in the ear. And then we give a challenge of that antigen. Usually that's given systemically. And you can see how much the ear swelling is induced. In this case, instead of giving the antigen, in this case again, peanut, systemically, we gave it orally. And we looked to see if we would see swelling in the ear. And as you can see, if you give serum that doesn't have IgE, you don't induce much of a response. And that's read out by this molecule Evans blue, which we can quantitate in the ear. But if you take B6 and C3H mice and give them both the same lot of serum that has IgE in it, only the C3H mice really have this swelling response, arguing that peanut was able to get out of the gut after an oral challenge into the systemic circulation and make it all the way to the ear in a form that the IgE antibodies could recognize. And so that's really the first demonstration of potentially a very interestingly unexpected deficit or change in the C3H mice that might be the cause of the susceptibility of this strain to oral anaphylaxis. And so we looked a little bit deeper, more closely now at the gut response. This is from a beautiful recent paper from Simon Hogan and Rod Newberry, who had originally described multiple ways in which antigen could access the systemic circulation from the gut lumen. And what they described were things that have been numerous names, but in one has been called a goblet cell associated passage, or a gap. And what they're showing in this image is the ability of these goblet cells, which are here in green, to pick up food antigen that was given orally in milk. So what we did is we just stained for goblet cells in the guts of C3H mice and C57 black 6 mice. And the way you can identify these cells is through a particular stain that stains very dark purple. And you can see there's many more purple dark staining cells that look like these passages in the C3H mice on the left versus the B6 mice on the right. And so we think this may be one way in which antigen is accessing the systemic circulation after an oral feeding. So this is our model of putting this all together. This is obviously still work in progress, and we're trying to understand both the genetic and molecular causes of what we're observing. We're thinking that the C3H mice are revealing a fundamental pathway for allergen to access the circulation, which is really a fundamental step for being able to access IgE on mast cells, which, as we know, are one of the culprits in inducing this anaphylactic response. And so on the top here, in C3H HEJ mice, when you have peanut coming into the gut lumen after feeding, it's in some way allowed to get through that barrier, just here the gut epithelial barriers shown as this wall in red, and it's able to get to these mast cells and induce degranulation and anaphylaxis. But for some reason in B6 mice, this is prevented. And so that may be because the epithelium are tighter, there are fewer of these gaps. We don't know. We're trying to figure this out now. So with that, I'd like to stop and first of all, thank my lab, which is shown here with our cool swag from one of our recent retreats when we could all be in person together. Thank you very much for your attention.